I'm Vivek. I'm going to talk today about Michael said. I built an application to enable the study of rare disease to form hypotheses and also to produce AAV-based predicted therapeutics off of them using a reinforcement learning framework. A bit about me. I have about 20 years background in drug discovery at Genentech, 10 years working in the guts of antibody engineering, and then 10 years working in the guts of research biology, thinking about how do you form hypotheses at scale. And it's interesting. You know, I think there's this problem we face in biology that you're probably familiar with, is that when you're going after an objective, it's never one objective. There's multiple objectives you're going after, and they're competing in tension, right? So if you're thinking about a hypothesis, it's not just the best hypothesis. It's the best hypothesis and the one that can be targeted by a drug. And trying to find that solution space that threads the needle, that's called a Pareto-frontier. And that applies to hypotheses, it applies to drugs. And so the question is, how can we apply Pareto-frontiers in drug discovery? And in fact, we do it all the time. And I actually think that's the center of a big fight that's happening in drug discovery today. And this really is between traditional biologists who believe that their intuition and their experience is what should drive the focused finding those solutions at the Pareto frontier, while more computationalists and machine learning scientists would argue that, well, your expertise in intuition is actually a form of bias. And so how do we thread that needle? I've seen this happen in real time. Probably some of you have too. And to illustrate that, I think the answer is they're both right in different ways. I mean, the reality is biology is a combinatorial problem. When you move from variant all the way to observed phenotype, you have a one-to-many relationship across the entire way. So you can have thousands of permutations or millions, depending on how you're proceeding. And the truth is, humans do expose. They go right to the center of the solution space they're familiar with. And that tends to lead to familiarity bias. And it's also true that machine learning scientists do a pretty good job of exploring the space, but a lot of what they get is garbage. And that's what produces this key tension. And I think the answer is you need to be able to thread the needle through attention, which we commonly use in ML, but I think is especially applicable here. And that is, I think it's very helpful for machines to do the exploration of design space and to guide scientific focus in intuitive ways to hypotheses, to variants, therapeutic variants that might be relevant in a disease context. And so what I've been doing over the last 100 days, I've taken the opportunity, because I know some rare disease researchers, to think about defining and building a prototype platform that allows us to do this in the context of rare disease. Rare disease is especially useful to ML because there really isn't good, there aren't great solutions in place to help patients, so they're highly engaged. And so you have a very engaged patient community and clinician community to work with. And so this is the basic therapeutic platform. We're applying it to a different rare disease, not the one I'm showing you today because I can't talk about it. But it's, the basic idea is we are formulating personalized disease hypotheses using, in my case, synthetic patient data. And then along with those disease, formulating personalized AAV treatments. So AAV is a kind of genetic engineering therapeutic. It's basically a virus you push into someone that mutates their genome in specific cell types. And then using both clinical data and validation assays as a reward mechanism to figure out like what to optimize. And so the focus I'm going to show today is something called DMD, which stands for Duchenne's muscular dystrophy. It affects males, children, and we're trying to understand both the mechanisms by which pathogenic variants affect the disease phenotypes, and also what are the AAV therapeutics that are optimized to treat this mechanism of disease. I'm going to show you a quick demo of this platform in real time. So you're seeing this Pareto frontier. This is for hypotheses. You're seeing this, this is for the hypotheses. Hypotheses that are designed for the strength of AAV discovery, and also what the hypothesis strength. So which of these hypotheses are both good in terms of evidence and also are likely to be targetable by a virus. This is the key thing you're trying to focus on. And what you're seeing is all of the precomputed relationships across the levels of biological organization, from variant to protein to pathway to subset or localization. These are precomputed images that showcase wild type versus truncated mutations. You can also see the subset or localization. And then as you drill down, we actually show you the actual AAV therapeutics designed for this person as well. Patient specific. Yeah, and these are the specific capsids we predicted that will be useful in the context of this hypothesis. So this is one part of it. We're showing clinical evidences here that informs this hypothesis. So that's one of the key things that's really important is we have to find a way to push clinical data into these platforms that we're building so that we can actually use this to actually inform patient readings. So this is now the other side of this. This is a Pareto-frontier exploring the AAV therapeutic side, and what I just showed you was the actual AAV therapeutic, and then this is providing the evidence. I'm trying to optimize for two contexts here. I'm trying to avoid neutralizing antibodies, so I'm trying to escape from neutralizing antibodies, and I'm also trying to maximize the chance of transduction into the right cell type of interest, and so we're optimizing for those two objectives. Now, the other side of this is attention. How do you build it to be intuitive to users? So this is what I'm using my rare disease collaborators for, is to build dashboards to view it and to be able to explore it in some detail. That's a high level. I'm happy to show you a demo if you'd like in more detail. So what drives this is a reasoning-oriented architecture. The reason I can scale this out is we pre-compute the representations that the agents need in advance. So agents aren't doing the work of trying to do all the data preparation. The data is just there in the format they need, and they, boom, can formulate these hypotheses and can formulate these predicted therapeutics. And that's what allows the scale to unlock. And so this powers two world models. One is an AAV-based therapeutics world model. That's a combination of data and protein models that are combining to actually make the predictions of AAV therapeutics. And the other side is a world model to generate the mechanistic hypotheses. These are all the data and models that are used to be able to provide the chain of evidence from the pathogenic variant all the way to the observed phenotype. So this is what my collaborators and I are building actively. We're actually focused on this right now. And the basic idea is we need a rare disease translational infrastructure platform to be able to meet the needs of the community. And this is what we're trying to actively ship and roll out, just as because we think it's the right thing to do. So I built this because I care about making a difference in the disease landscape and trying to contribute as best I can. And when you learn about the disease, you'll want to contribute to. It's pretty heartbreaking what's happening to a lot of people afflicted by these. So in the next 90 days, we're working on QCing this framework that I built out, rolling it out to my rare disease research trust in the disease context they're focused on, integrating new analytic workflows into the framework to make it more accessible and intuitive to them, and then building a collaboration with a sample management org to actually try to get some of this data. And with that, I'd love to collaborate. If you're a researcher and you want to talk to me, I'd love to chat with you. If you want to help build or extend this, let me know. I'd love to collaborate. Or if you want to help fund and create this platform, that would be great too. It's not cheap to do something like this. Tokens are expensive. And with that, I want to say thank you to Michael and this whole Build Fellowship been a part of. I have to say, it's one of those things I recommend you try to do, even if you don't feel like doing it, because the act of making something makes you deeper and it gives you more confidence to explore that area. So I highly recommend it. So thank you all, and thank you all to my fellows. And that's it.